Student, today we are going to start a new topic of genetics, which is translation. We have discussed about replication and transcription, which was the part of central dogma, and today we are going to start a second part of central dogma, which is translation. Translation is actually the formation of protein from mRNA, which was made during transcription. Now the cell need to make protein for different functions, as protein plays very important role as a structure, as a function, and many enzymes are protein in nature, and that are very crucial for the mechanism and for the metabolism of organism. Translation is will be occur inside the cytoplasm, while the replication and transcription was done inside the nucleus. So once the replication and transcription was done inside the nucleus, the mRNA which is made during transcription have to move out from the nucleus for protein synthesis, because the ribosomes which help in translation are present inside the cytoplasm. And ribosomes are single non-membrane bonded organelle which is present inside the nucleus, and there are huge number of ribosomes inside the cell. Ribosomes are actually dispersed inside the cytoplasm, or maybe are present on endoplasmic reticulum, and on that part of endoplasmic reticulum which contain ribosomes are called rough endoplasmic reticulum. As we have discussed, transcription and replication always proceed from 5 to 3 direction. So translation will always proceed from 3 to 5 direction. As the polypeptide chain, the first amino acid of that polypeptide chain contain free amino acid, so that end will be called N-terminal, and the last amino acid of polypeptide chain has free acid group, so it is called C-terminal. As we have discussed in genetic code, there are 3 nitrogenous bases that form one codon, and one codon always code for single amino acid. So one codon will always code for single amino acid, but one amino acid could be have more than one code. So the RNA are basically 3 types. The first one is mRNA, means messenger RNA. Second is transfer RNA, and third is ribosomal RNA. Here we will discuss about the transfer RNA first. Transfer RNA molecule are short RNA that fold into a characteristic clover leaf pattern. Some of nucleotide are modified to become things like pseudo uridine and ribothymidine. Always keep in mind the transfer RNA always contain anticodon. The function of RNA, the function of transfer RNA is actually this transfer RNA will read the code of mRNA. mRNA contains specific sequence of nitrogenous bases. It will read that particular pattern of that amino acids, and then it will search out inside the cytoplasm the opposite of that codon, as the mRNA contain codons and transfer RNA will contain anticodon. It means, for example, if on the mRNA is AUG, means adenine, uracil, and guanine, then always keep in mind the in front of adenine always thymine bind, and in front of thymine always adenine bind. Okay, so the mRNA contain codon and the transfer RNA contain anticodon. Each transfer RNA has 3 bases that make up the anticodon. These bases pair up with 3 bases of codon on mRNA during translation. Each transfer RNA has its corresponding amino acid attached to third end. A set of amino acid, the amino acyl transfer RNA synthetase, are used to charge the transfer RNA with proper amino acid. Some transfer RNA can pair with more than one codon. The third base of anticodon is called the Wobble base, and it can form base pair with several different nucleotides. It means Wobble base means that can base up with several different nucleotides, and the third base of one anticodon is called Wobble position. Then the process of translation and initiation of translation, how it could be taken place inside the in prokaryote and eukaryote, the process of translation is somehow different, but most probably the mechanism is same. In first, we will discuss in prokaryote. In prokaryote, the ribosomes bind to specific translation initiation site. As a first step is initiation. There may be different initiation site on messenger RNA. A prokaryotic mRNA can code for several different proteins. Translation began at AUG codon, or maybe it can start at GUG codon. As we have discussed in genetic code, there are 64 codons, and out of that 64 codons, 3 codon are nonsense codon that actually stop the translation process, and the one codon is a starter codon that actually start the translation process. So AUG codon is a starter codon that initiate the process of translation. The modified amino acid N-formyl methionine and AUG actually code for amino acid which is methionine. And methionine is the first amino acid which is present on the polypeptide, new polypeptide. In eukaryotes, ribosome bind to 5' and as you know, before moving the mRNA from nucleus to cytoplasm, its capping and tailing was taken place. So the enzymes which is present inside the cytoplasm cannot attack on that mRNA and cannot destroy them. So the modification was done, and the modification was on 5' there was capping done, and on 3rd poly A tailing was done. So in eukaryote, the ribosomes will bind with 5 cap, then move down the mRNA until they reach the first AUG codon, which is the methionine codon. Translation start from this point. Eukaryotic mRNA code for only a single gene. Note that translation does not start at the first base of mRNA. There is an untranslated region at the beginning of mRNA, the 5 untranslated region, 5 UTR. Then more initiation. The initiation process involves first initiator transfer RNA and the small ribosomal subunit. Several initiation factor, additional proteins are also involved for the formation of initiation complex for the start of protein. The next iteration, initiation